with the stage of cancer, with the increase in invasiveness of the disease, this methylation was found to be increasing. If if we're able to detect the hypermethylation in the 1A stage, suppose, the methylation specific band was, not that intense. But as we moved on with the stages, suppose I had, 5 4B cases. In those, the intensity of methylation specific bands was very high, and when we correlated it with reverse transcription PCR, the gene expression was almost silenced. And when we, went for quantitative real-time PCR also, this was completely, a similar story. So present study indicated that, these genes, as I told you, P16, P73, RAR beta 2, FHIT, and DAPK, these genes are significantly hypermethylated in North Indian population in cervical cancer patients, and the hypermethylation of these genes is a common phenomenon in cervical cancer and may be an important way to inactivate tumor suppressor genes in cervical cancer. Now, we wanted to correlate the methylation status of these genes with HPV infection and stage of cervical cancer. As I already told you, percentage of samples showing methylation of these genes, RR beta 2, P73, FHIT, and DAPK, was found to increase with increase in stage of cervical cancer. In stage 4B, the methylation was observed in most of the cases studied, and the gamma coefficient was found to be significantly, important in case of all these genes. Like P73, it was found to be 0.352, and the p-value also was found to be significant. You can see this. Now, these were some of the things how we, did the RNA isolation, DNA isolation, all these details about all these families of genes which we studied. And this is the papillomavirus life cycle and how it induces cancer in stratified squamous epithelium of uterine cervix. You can see how it infects, how the productive cycle is carried out, and how E1 and E2 are expressed. These are oncoproteins, and persistent infection leads to neoplasia, and then precancerous stage, and followed by the cancer of uterine cervix. These are my publications which are related to this work which I have presented here, and these are the abstracts published by my group in various conferences, national as well as international conferences. And these are my collaborators: Dr. Anju Srivastava, Professor, Department of Zoology, Delhi University, Delhi, India; Dr. Jagdeep Kaur, Professor, Department of Biotechnology, Punjab University, Chandigarh, India; and Dr. Anshuman Kumar, Director, Surgical Oncology, Dharamshala Cancer Hospital and Research Center, Delhi, India. Although I've named only a few, I had, I was really lucky to have many more collaborators like Dr. Veena Jain from Mohandas Oswal Cancer Research Center, from where I got the ethical clearance and where our group, got the samples for, cervical cancer patients, as well as, Dr. Alka Sehgal. She was very important. She was the director of, the Government Medical College and Hospital, Chandigarh. And there were several other people like Dr. Neena Kaplash. She was my co-supervisor at Punjab University, Chandigarh at that time when I had started the study, and I carried on this study even after completing my PhD. I carried out for 7 more years. So with my students, and I'm having 4 PhD students. Out of them, 2 are working on this aspect, and 2 of them are working on liver cancer and oral squamous cell carcinoma. So it has been a long journey working in the field of cervical cancer. I nearly worked in this field for nearly 10 years, and nowadays I focused my study on oral squamous cell carcinoma as well as liver cancer, and we are also trying to see how the contaminants present in groundwater, they are causing the epigenetic changes which are resulting in cancer, because at several places across the world you must have seen that there are certain inorganic as well as organic components which are causing epigenetic changes which are resulting in various kinds of cancer. And then we are also trying to look upon the various aspects of Alzheimer's disease, which is a progressive case of progressive dementia. So that is the thing. And with this, I would like to say thanks to the organizers as well as all of you, the audience, for patient hearing. Thank you. I would be happy to answer any questions. If you have any questions, you can please ask me. Thank you. Thanks a lot.